I would like to thank Sarah for organizing this session on COVID-19 question and answers. But before we take question and answers, I thought I would go over some of the known or things we've learned about SARS-CoV-2, the virus that causes COVID-19 disease. So with that, I will start some of our discussion on COVID-19. Let's see. So I thought we'll just start with context and a bigger picture. And of course, this is not the first pandemic that we have experienced. And as you can see, if we were to look at the death toll from the other known pandemics, COVID-19 is right here, which so far, of course, is difficult to come to the numbers or exact number of deaths due to COVID-19. But as you can see, there have been other pandemics that have caused a lot of havoc. And the Black Death, of course, with 200 million people dying. Now, of course, my question, I tried to figure out how did they ever calculate how many deaths happened with these pandemics in the past, and especially in pandemics that happened in the BCs, but I couldn't quite figure out how this was calculated. But let's just say at face value, if you believe that these are the numbers, then that's where we are in terms of where COVID-19 stands. So then I also looked at the CDC and WHO. These four institutions have pooled their resources and put together this website where you can track deaths from different diseases, including COVID-19, in real time. And so I have from here, from yesterday, since I was preparing my slides, the number of COVID-related deaths in India. And as you can see, it shows me about 318,000. So COVID is somewhere number 12 in number of deaths, with heart disease being really on the top. And compare that to Canada, where my host is, and as you can see, COVID is number 4 leading cause of death, but the numbers, if you compare absolute numbers, they're absolutely, apple to oranges. Mm-hmm. So between yesterday and today, I looked at what happened in India. And as you can see, it showed me that between yesterday and today, there were about 3,600 COVID-related deaths, but there were 4,400 heart disease-related deaths, and then there is also flu-related deaths. So just keep that in perspective, that there are other problems that are pretty significant, and we've gotten so distracted with COVID and so consumed with COVID that I'm not sure where we'll go with that. So let's look at some other stats. And there are people who join in from Australia, and as you can see, in Australia, a number of COVID-related deaths are very, very low, and I'm sure New Zealand is the same, and it, in fact, is at the bottom of death-causing diseases. But in the US, over here, and in the UK, COVID is on the top. So this is despite all the social distancing, despite all the precautions we've been taking compared to what's happened in India, where social distancing is somewhat not quite achievable for most of the people, and the population-wise, it's much more dense. Area-wise, it's 1/5 of the U.S. Area. So we still have a lot of difference in terms of how much of a havoc COVID-19 has created in the US and in the UK. And what's happening here is basically what drives policies and thinking in the rest of the world. So we need to think whether or not in places like Australia and New Zealand, where there is not that much of an impact, and yet there are pretty draconian measures in place. So what is COVID-19? It's, as we all know, is called SARS-CoV-2. And I took this from a recent review by somebody who's actually based in Australia, and it's a pretty comprehensive review. The main thing really is that these family of viruses, the SARS viruses, natural hosts are bats. And they've been studied extensively in China, the horseshoe bat, or in Asian countries. And most of the time there is a species barrier, meaning the viruses that you find in bats will normally not be able to replicate in other animals, including humans. So this here remains still a black box. How did this virus jump from bats to humans? And there have been thoughts that perhaps it jumped into an animal called pangolin, and then from there into humans, but it's still not clear as to how this particular SARS-CoV-2, the only SARS virus or coronavirus that has this furin-like cleavage site in its spike protein, which is the protein that's used for making vaccine. And what is curious about this is that furin is an enzyme found only in humans. So how did this virus acquire something which is only present in humans without first encountering or having some interface with either human cells or cell lines? So this indeed remains a black box. And of course, there are all theories floating around whether it's and natural is really the operative word here, meaning is it really a natural mutation which allowed it to jump from bats to humans, or is it that, and it's not the first time that things have leaked out of the lab, that you're doing experiments and you have certain mutations inserted into viruses or bacteria, and they escape, or people in the lab working get infected. And so that's another possibility. And I do not think we have enough data to rule out any of that, and especially in absence of things that we don't have any way of knowing how a bat virus, which has never seen something like this, acquired that. So that's something to keep in mind. But nonetheless, here we are. COVID is now rampant in human population. And as is with the family of these viruses, you get a respiratory infection. But unlike other coronaviruses, the other sort of unique thing about this virus is that it's not just the respiratory tract that it seems to infect. It seems to infect a number of different cell types. So it can infect gastrointestinal cells, it can infect brain cells, cardiovascular cells, or heart cells. And one of the reasons, and in fact, I found a couple of old papers where they showed that if they take this virus and they make it go through human cells from these different tissues, over time, it can acquire this polybasic furin-like cleavage site. And once it does that, then in those cell types, it's really very infectious and it will kill those cells. So for some reason, this particular virus, because of its harboring this polybasic sequence in its spike protein, it seems to infect more organs in humans compared to the previous SARS or the original SARS that happened back in 2000. So in a subset of people, of course, this infection is not cleared, and it becomes more of a respiratory distress syndrome with oxygen levels falling below certain levels, and in some people there's thrombopenia, meaning lysis of blood cells, and eventual organ failure and death. So there's obviously been a lot of debate about there are a number of people who are either asymptomatic, there are people who are symptomatic but have very mild to moderate symptoms, and then of course there are people who have very severe symptoms, which leads to pneumonia, and then all the other things that we talked about. But I think there is enough evidence now that people, whether they have mild to moderate disease or symptoms, they still generate a robust immune response. In fact, the studies done from Singapore, the studies done from other places where they've shown, and those titers are pretty high, and that's where we are with that. So I think people who've had COVID, they have a pretty high titer of antibodies, which can neutralize subsequent infections, and therefore you may not see those subsequent. The CDC website gives some information about, or a lot of information about SARS-CoV-2, and next few slides I've just taken off from the CDC website without largely modifying them. But so this talks about that there are multiple variants, as we know, and these variants are constantly changing. Unlike other flu viruses or other viruses that we've known, it seems to be that these mutations are appearing at really a much faster rate than we can even count. And some of these variants, they after emerging have disappeared, and others have persisted. So the problem with some of these variants are that they are more transmissible, meaning they can spread much more easily from people to people, because that's what the virus is intent is to survive without really killing the host. But just because it's more transmissible doesn't mean it is more pathogenic or it causes more severe disease. So in these new variants, they can cause either milder disease, or it can cause more severe disease in a subset of people. And we need to adapt our diagnostic tests to detect those. And of course, PCR, RT-PCR is one of the more reliable sources. Antibodies are much more difficult to make against mutants, and so you can miss them. So, and then of course, the biggest worry, as was sort of mentioned before, is that are these mutants then, or are our current vaccines even effective against these? So one thing to remember, antibodies are like, they're made to a shape, more like, you know, your hand fitting in a glove. So if your hand, if it's not my hand, it's not my glove, it may not fit. Or if we are a very similar size, it may fit, but it may not be a perfect fit. So when you have these mutations, the shape of the protein changes, and therefore the antibody may not recognize that particular mutation. So these are the variants that CDC has posted on its website. People have heard their names or what these mutations basically are. These are all amino acids that are that have been changed. So, for example, here you have serine at a particular amino acid residue, which is 477, changed to an SPAR gene. And some of these, you know, they may not make sense to the general public, but again, some of these are what are called polar residues, and some are non-polar, some are hydrophobic, some are hydrophilic. And depending on what these mutations are, the way the protein folds and the spike protein is then going to look is going to be different, and therefore it may or may not be recognized by the antibody. So there are some variants that CDC call variants of interest, and then some variants which is called variants of concern. And it's calling them variants of concern because these variants, they are more transmissible. So they are obviously so different from the original way the protein is made that the vaccine may not neutralize it, or it may have a reduced efficacy. So of course, we don't know what % of people harbor these kind of mutations. So what is really intriguing to me is the rapidity at which this virus has mutated, and the number of mutations that have already existed in less than a year, which of course makes it incredibly difficult to make a good vaccine with an antibody that can neutralize all of these different variants. So let's talk very quickly about the different kinds of vaccines that we have. And traditionally, so some of these, of course, are recommended in the US, which are on the top panel, and then on the bottom panel are vaccines that are suggested that people take in the US, but they're not sort of mandated, if you will. So the different kinds of vaccines are, you can have a live or attenuated vaccine, which means you take the virus and use human viruses or viruses that cause diseases in humans, such as measles, mumps, chickenpox. You take them and you grow them in a different host. So in most cases, it's chicken eggs, and you grow them several times. So you passage them 10 times or 12 times, and by the time it goes through those different cycles, it basically forgets that its host was human, and it loses its ability to replicate then in human cells, and now will replicate in chicken. And therefore, when you take the virus and give it back to humans as a vaccine, it can no longer replicate in us, but it can still evoke an immune response, and therefore you develop antibodies, and you develop long-term antibodies, so you remember them, and therefore they're pretty effective. Then there are other kinds of vaccines, which are you either heat inactivate or you chemically inactivate and kill these viruses, and then you give it to humans. So of course they're killed, so they can't replicate. Or you can make, you can take in terms of especially bacterial toxins, and then you inactivate those toxins, and then you can get vaccines against them. And tetanus is one example. Or you can make sort of recombinant vaccines where you combine them with some other virus, which is adenovirus or some other virus, and then you will get a combination of that particular virus as well as the virus in question here or the bacteria in question, and then you get vaccines against that. Important thing to remember is that if you're taking a live virus and you're passing it through some other host, such as chicken or dog cell lines or whatever, and it's then losing its ability to then replicate in human cells, the converse is also true. If you take something that's the virus whose natural host is some other animal, and you put it in human cell lines and you go through different passages, then that can acquire its ability to replicate in human cells. So the chickenpox vaccine is a good example. It's known to be pretty effective, but we still see some breakthrough infections. So chickenpox, if you've naturally had it, it's very, very, very rare that you will get it ever again. There are cases or incidences where you could get it again, and that is because if you had chickenpox, if you were younger than 6 months old when you got it, or when you were older but your symptoms were very, very mild. That's the only time when you've had naturally chickenpox that you can get it. Otherwise, it's impossible to get a secondary infection with chickenpox or a second-time infection. But if you've been vaccinated, it's, as you can see, different people report different efficacies, but overall, it's still 72% efficacious. But what was really interesting is that if in this particular outbreak that happened, which was reported in 2002 in the US and Michigan area, they found that if the children that were vaccinated for more than 5 years before the outbreak, they were at greater risk of getting the infection than more recently vaccinated kids. Coming to the SARS-CoV vaccines. So initially there were 48 vaccines that were started in phase 1, and it takes a long process. And in phase 3, there were still 27 that were tested for efficacy. And so far, we have in different countries a total of 8 vaccines that have been approved for emergency use authorization. So that's the operative word, meaning it's approved, but it's still under emergency use. So we still don't know to its full, the full adverse effect of these vaccines or its safety. And we need to remember that the efficacy, the way they've defined what the efficacy is really, is to say that if you, that it's basically decreased incidence of infection. And the secondary outcome is reduced severity. Nowhere do they, any one of these vaccine makers claim that it's going to prevent complete infection. But we put words in their mouth and we assume that these vaccines, basically, if you take these vaccines, you're completely immune. But whereas none of these companies are claiming that that's what's going to happen. All they're saying is that it's going to reduce the incidence of infection and perhaps reduce the severity of infection. So how do you get protection from these vaccines? Like we talked about earlier, that we have antibodies that recognize the shape of the spike protein, and so it binds to that protein and basically neutralizes or degrades it or kills it or removes it from your system. So now if you have that mutation and the protein is not folding in the manner that these antibodies recognize them or bind them back to lock or key or hand in glove kind of analogy, then it doesn't really work as well. So then you end up with these mutations that are called escape mutations, which do not, these antibodies or vaccines that will not be able to recognize or neutralize. So again, where we stand as per CDC, correlation of protection or threshold that protects against SARS-CoV-2 has not yet been determined, meaning this, how much of antibody do you need to have in your circulation that will give you protection? It's still under question or still needs to be determined. And of course, the variants, they result in reduced protective antibody level, and we still don't know what the duration of protection from the vaccines or even natural infection, for that matter, is. But having said that, you, again, very rarely hear of people who have had COVID with some moderate to severe symptoms, those people getting COVID again. You might have heard of people who might have tested for PCR positive, even though they had COVID before. But again, the virus, we don't know how long it can linger, or it may hide in certain immune cells. And since the sensitivity of the technique can be pretty high, or it may be even just a false positive. So we don't know that. But again, there are enough number of people who are fully vaccinated but still have breakthrough infection. Finally, I would like to just point out that even science is not perfect and there are lots of flaws, and scientists are humans and they also like catchy phrases and catchy headlines. And so this is an interesting study that just came out which showed that research findings that are probably wrong are cited far more than robust findings. So, and that is because more robust findings, people who report them, they don't make very sexy headlines, whereas data that is perhaps, or we want to see it as more attractive, makes these kind of very attractive headlines. So this is the original paper, which says non-replicable publications are cited more than replicable ones. And I'm sure most people who would see this would probably ignore it. But if you see a catchy headline like this, you would definitely gravitate towards it. So the other sort of misconception that seems to be floating around is that COVID is unique, that it's causing this brain fog and dementia, or it's causing these long haul symptoms, and people are like, however, COVID-19 is only one of many infections, from Ebola to strep throat, chikungunya, dengue, malaria, that has persistent symptoms in some subset of patients. And as one of the immunologists from Yale says, that while there's no doubt that long COVID is a real condition worthy of diagnosis and treatment, this isn't unique to COVID. And another microbiologist says that she would be surprised if COVID actually didn't cause chronic symptoms, because if that were the case, it would be the only virus that didn't do that. So again, let's keep our perspective and put things in context. So finally, this is one of the latest papers that has come out, which sort of claims the vaccines are just as efficient as a natural infection in neutralizing antibody. So I want to just take a few minutes to walk you through this, because I want you to understand how, if you don't, even for scientists, it's really hard to digest the information that's presented in these kind of papers, which are really complex papers, and you need to read through them maybe a number of times. Maybe you will need to find information that will tell you whether or not you can take the data at face value, or you need to view it with some caveats. So here is an example. It's a highly sought after journal called Nature. And this is the title of the paper. And I'm only taking part of the figure to show you what they've done here. And what they've done is they recruited 20 patients who were vaccinated. So that's N equals 20. And in the black dots represent people who were vaccinated with the Moderna vaccine, and the red dot represents people who were vaccinated with the Pfizer vaccine. So if you break that down, it's only 14 and 6. Now, it's a very, very, very small number. So if we are going to take this as gold standard, it's a problem, in my mind. But based on this paper, most people, including CDC, has put on its website that the vaccine is very effective in neutralizing the antibody. Now, what antibody are we talking about? This is, again, a man-made antibody that we are testing, which is a pseudotype of the virus packaged in HIV virus to make that antibody, and that's what is being tested in the lab, which is it's neutralizing. Nobody's actually looked at whether or not the naturally occurring SARS antibody can be neutralized by vaccinated patients or people who've had COVID. So here are people who've had COVID, and their N is much more robust, perhaps 89. I couldn't find it in the paper. I had to actually sit and count these dots, and some of them are overlapping, so I may not have done a good job. So with that disclaimer. Now, the other problem or sort of disparity is that they recruited patients who had COVID, and they got their blood either after 1.3 months of infection or 6 months of infection. The vaccinated people, they don't write it. I have written it over here, were recruited within 3 to 14 weeks of infection. So it's expected that their titers are going to be high. Or that means they will have a naturally high level of antibody if the vaccine worked. So then they say that an average of 8 weeks. So that will look closer to 1.3 months. But the truth is, you have such a small number, an N of 6. They all could have been just 3 weeks out. We don't know that. It's really difficult to find that in this paper. So with that caveat, I would say that people who were naturally infected, they have a pretty good chance of not being reinfected. And based on that, the authors conclude that there is no significant difference in neutralizing activity between the Moderna and Pfizer vaccines. But look at the N's here. The second important finding based on which people have made claims that there's going to be long-term immunity from vaccines, which is this particular experiment where they look at these kind of immune cells called memory B cells, and they find that compared to vaccinated people, people who have had COVID, their number of B cells that can recognize or have persistent memory for recognizing these antibodies is pretty similar. So based on that, they have made the claim that vaccines will give long-term immunity. But on the other hand, so will getting it naturally. So, but this again is a very small number. So with that, again, CDC, including, CDC's reached that conclusion that neutralizing antibodies in sera from mRNA vaccine recipients is very similar or better than COVID-19 convalescent sera. I do not completely agree with that conclusion. They do go on to say that all these variabilities can be explained because of different methodologies. Of course, we don't know when. They don't tell us how different kind of post-vaccination times are not known. And most importantly, they do state that the sample size are very small and you cannot generalize. And yet here we are generalizing from these small sample size studies. And the other thing that they point out is almost half of the studies are preprints. They have not been peer-reviewed. So yet we are taking this as gold standard. So I would say that science should not be consumed in real time. It's constantly evolving. And really the answer depends on the questions you're asking. The controls are very important. So with that, I'll stop. And
thank you, Aditi. This was amazing. You confirmed a lot of my questions anyways, but I'll open it to everyone else. Yeah, please go ahead. I don't know in what order, but we'll see if come up with your questions, or maybe you can answer the question that was already asked, that do I need to take the vaccine if I don't want to travel?
I guess if New Zealand doesn't enforce or require you, then of course, I think again, it's a personal choice. I can tell you personally, I have not taken the vaccine and I still think it's an experimental drug. I feel that I am pretty healthy. I trust my immune system, and it's a little bit like saying, immune system needs to be exposed to immunogens or antigens, or otherwise, how does it get trained? To say that vaccines are equivalent to natural infection is incorrect. And we have Mukta here on with us, if she's still here. She is an expert immunologist and working in Oxford, and she can perhaps better answer that.
So Mukta, are you there?
So it's a little bit like my analogy over there is to say, let's say I get trained in karate and I'm a black belt, and then I don't do anything for next 5 years. Am I going to remain a black belt level? No. You look at all the athletes; they have to constantly train. If they sometimes even if they miss a few months of training, they're not. So Mukta saying she's unable to unmute.
Okay.
Let me see if I can.
You can unmute her. Next time we do something like this, we need a special technical staff.
So going back to that analogy, if you didn't train, then you're not going to be able to keep up.
So that's great. And I think the other question was, there are people who don't want to take the vaccine, but they are being forced by their friends or family who have taken the vaccine, and they're being told that you are a threat to the society if you don't take it. And how would you kind of help those people tackle that question with their friends and family?
Can I just say something before you answer that, Aditi? Because even with your, I'll put my video on, even with your analogy about training your system, that's fine. But I'm going in to take care of my 93-year-old mother who doesn't have the luxury of training her immune system. So I got vaccinated so I can protect her. So I think for me, the argument is always, I mean, we've had these discussions, but the argument is what's good for me is not necessarily good for my community and my society. And I think that that's why I've had the vaccine, is because I am trying to protect the people around me. I think I probably could fight it off. I honestly do. Or get sick and build my own immunity against it. But I can't take that risk when I'm taking care of my 93-year-old mother and the people in her building. It's just, and I think I even said this to my kids because we had the same arguments. Well, if I get it, I'll be fine, and then I'll have my own immunity. And I'm saying, it's not about you. It's about the society around us and protecting the vulnerable.
So I think that's it's a personal, in some ways it's a personal view or choice, to keep in mind that when we talked about the virus, when it goes through healthy people, it has the potential of becoming less potent. Whereas if it only goes through people who are more sick, and or people who are for some reason unable to take vaccine, and they are still, you can't guarantee or protect them, or unless you're putting them in a bubble. And so if the virus goes through them, the potential of it, becoming something worse is more likely. So having that controlled population that's not been vaccinated, who is healthy, who can take that hit and perhaps make the virus less pathogenic or less virulent, is probably, I think, is a smarter approach. Again, and I think the fear and the misunderstanding or the misconceptions that have been put in people, I think it's true for even with the flu vaccine that's been around. Really, if you look at the data, its efficacy is only 30%. And, we haven't gotten rid of flu. We haven't gotten rid of, these are RNA viruses. They're not like chickenpox. They're not like measles. They're not like mumps. These viruses mutate. And as we've seen, as I showed you in that slide, even in this 1 year, it's mutated so often. So given that we don't know whether or not the vaccines will protect us against those mutations, I think people who are healthy and who feel that they don't have the same constraints as you have, Colleen, I think those people should be allowed to take that risk and become naturally immune. They will, in a way, protect the other people.
I agree with that. And again, it's a point of convincing people, like you said. The media and the sexy research has trained our minds. People who generally do not look at scientific literature are now having to kind of make those decisions, right? And they are having to learn very quickly, and it's confusing, and especially this whole concept of vaccinate everyone with an mRNA vaccine and then we're all going to be safe. That's I don't know how to break that myth. Maybe you can do a better job of that.
May I say something? Better off if we're getting it and then diminishing the effectiveness of the virus. However, at the same time, those who have gotten it for a reason or another, I think we should applaud that as well. Thank you.
And I do think that you can still take precautions such as wearing masks and sort of maintaining some sort of distance, because I've seen some studies where, even though with the caveat, I will cite or explain that study where they took a bolus of virus and spotted them on different surfaces, so on glass, on steel, on cotton, and other kind of materials. And they spotted quite a bit of the virus, 10 to 6 PFUs, and then they put them in the incubator, and then they took that material out 24 hours later, 3 days later, 7 days later, and 21 days later, and asked how much of the virus could they grow back. And in fact, cotton was one thing, they couldn't recover any virus from it within 24 hours. So if you, again, I think if the kind of N95 masks that I've seen around, it just says N95, they're not N95. The N95 masks have to be specially fitted. But having said that, just wearing a cotton mask may be able to help you.
I don't know. Someone said that they can't hear anything. Is everyone else able to hear, or I'm able to hear you?
I think you have to make the audio from your side, Kinney, louder.
Mm-hmm. Right.
And I think you should be able to unmute yourself because obviously Colleen is able to do that, Sarah is able to do that. So Mukta, on your pad or maybe somewhere on your screen, if you just scroll with your mouse, you should be able to see.
Okay, good. Okay. So I guess the question we are dealing with here is that people think that the vaccine is 100% effective, and I don't know. I mean, your presentation is very clear on that, and it's not data that you're making up. The vaccine has not been approved because it is still missing the safety data. Is that correct?
Right, but just remember that when they say, again, when they say it's effective, they're talking about decreasing incidence. And the jury is still out whether or not the incidence is being decreased. Partly I say that because, again, the way they did the data analysis is that when they gave the vaccine and they gave placebo, so the placebo arm, they call the people every week to test them, whereas the vaccinated people were called only once a month. So now you're comparing apples to oranges. You're testing one arm 4 times, whereas you're testing the other arm only once. So then to say that the incidence is decreased, you have to take it with a pinch of salt. Mm-hmm. But nonetheless, there is data that I just presented from this Nature paper that, to me, convincingly shows that in people, or at least a subset of people in which there are antibodies made after they've been vaccinated, they can neutralize antibodies or the COVID-2, the viral antibody, and therefore they may have a less severe disease. But again, the ends are very small. We don't know if everybody who's gotten the vaccine is actually making the antibodies, and I know a few of my colleagues tested themselves and they haven't found any antibodies. So it's possible that there are a subset of people who've gotten the vaccine, but they will not make antibodies, and we don't know whether they'll be protected or not. So again, in that respect, it's true that we can't just because you've been vaccinated does not mean that you are completely immune to it. And so if it gives you a false sense of security, then that's also a problem, unless you know for sure that you have antibodies after you've been vaccinated.
Right. And do you?
But I don't think none of the studies are. The studies are across the board. They told us in Canada that it will not decrease the incidence. It decreases the hospitalization and it decreases the deaths due to COVID. So I may still get it, but the level of severity of the disease is less. I don't think any of the vaccinations or companies are saying that they're going to decrease the incidence.
They are saying that it will decrease the incidence there, and the secondary measure is decreasing severity as well as decreasing the length of hospitalization. But their primary measure is actually decreasing incidence.
Well, I never heard that from any of the science.
Okay, I will send you the document that was presented to the FDA by Moderna and Pfizer. And I've highlighted in that the primary outcome is actually decrease in incidence.
Yeah, and maybe that, like I know Health Canada is quite clear, it doesn't decrease incidence. We are approving it because it decreases hospitalization and it decreases deaths from COVID. That's what their Health Canada has approved. They're saying we don't have the evidence that it decreases incidence.
Okay, I'm going to read some of the questions in the chat box. Sorry. So I will start with the easier ones. How long before world will be COVID-free? COVID is here to stay.
Just like the flu virus, right?
It's like the flu virus. It's here to stay. So is it man-made? So again, there's a difference between man-made, meaning it's not somebody's not sat down and actually designed every single nucleotide or the genetic blueprint. But like I said, what can happen is that if you, the people who've been studying these viruses from the bat, they use different kinds of cell line to grow them. And if you grow them in human cell lines, you grow them enough number of times, then they can acquire ability to grow and replicate in humans. And if then it gets from there, from, you know, you're working in, there are certain precautions one is supposed to take, and if you don't take those precautions, you can get infected. There have been you in the lab who died from studying infectious pathogens and things like that because they've gotten infected. So whether or not somebody in Wuhan working on this virus or some other place in the world working on this virus somehow got infected because they were growing them in human cells, and then got it from there, and then it mutated into something that was worse or less pathogenic, is the jury's out.
Can I ask a question, Aditi, please?
Yes.
This is Catchy here. In India, we are getting Covishield, which is an AstraZeneca vaccine, and we're supposed to take 2 doses of that. So initially, when they started vaccinating us, they said the difference between the first vaccine and the second vaccine would be 4 weeks. Then after some time, they started saying it has to be 4 weeks to 6 weeks. Then it came that it has to be around 12 weeks. So do we take it that the research is still going on and they're discovering new facts every day, and so this period, the interval between the 2 vaccines is increasing, or is it due to some other reason?
I really wish Mukta would unmute.
There are many who have taken the vaccine within an interval of 4 weeks. They already took that vaccine. I took it within an interval of 6 weeks, and the day I took it, the next day the finding came that it has to be taken within an interval of 12 weeks. So what is this? I mean, it's increasing, the interval is increasing every day. Is it new researches are being carried out every day and, new findings, or it could be something else?
I really wish Mukta could unmute herself because she would probably be better. Or maybe you can type some of your responses, Mukta, if you can hear us. But so the thing is that it's a range. And the second dose, in most cases, is given to boost your first immune reaction. And that's because, primarily because the vaccine doesn't work like the natural infection. You don't need a booster for your natural infection. When you get naturally infected, there are multiple steps that are involved in recognizing the pathogen, different components of the immune system that are involved. And the training that is done is very different than when you have a vaccine. So the vaccine normally is conjugated, or there are many things that go into making that vaccine, and to generate or to try and fool your immune system to mount a robust immune response. And so the booster is needed to do that. And so the duration between the booster can vary. And there is no, and I think what you asked, Raki, all of them are driving factors, perhaps unavailability of the vaccine, or just shortage of it, or, making sure that more people at least get some sort of immunity versus, I
think I've managed to unmute myself.
Yes, there we go.
So I had a small thing to say about the AstraZeneca one. From the little bit that I knew about the data was that although the initial trials were done at a 3 to 4 week interval, which is what the company recommended, because that's what the trial was done at, and that was when it was approved, that was the emergency approval. But there's been newer data that's been subsequently analyzed that seems to suggest that, because in the initial trial that they did, there were multiple batches that they did, and when they unblinded the final stages of the study, those data were done where they had a longer interval. And so there's been a little bit more evidence, and I don't know if this has been published as yet, but I've seen these in this evidence in seminars, so it's not peer reviewed, that the longer duration between the first and the second dose seems to boost the titers of neutralizing antibodies. if that's this is specifically with the AstraZeneca Chadox vaccine version. That's about the data that I can. So here on the NHS here in the UK, they have been right from the beginning giving these vaccines, the first and the second dose, on a 12-week interval. So I think India started off on a 3-week, which was what was recommended by the company. And then I'm guessing it's a balance between public health, vaccine availability, and as data comes through about whether... I had a question, actually a couple of questions about the vaccines, because I haven't really seen a lot of data about the RNA vaccines. One was whether there is anything to suggest that I have seen data that suggests that there is decrease in severity and hospital. So people who've had severe infections, that the chances of surviving, so mortality reduction, there was some data. And obviously there isn't any real support for protection from getting the infection. So that I think is quite... But I was wondering whether there was any data on reducing transmission, because that would be relevant. Whether an individual is infected, but whether in the community, whether there's reduced transmission because of the vaccination. And my second question to you was, because again, I haven't seen the trial data, was you said that between individuals who had had the vaccine and people who had the placebo, there was differential sampling. But that would suggest that it was not a blinded study. If that was the case, is that actually this? Because I know that the Chadox study that happened in Oxford, I know individuals who were on the vaccine in the trial, and it was a blinded study, and they got swabbed, PCR swabbed literally every week. So I was wondering whether the Pfizer and the Moderna trials were actually not blinded, because that would be a breach, right, if their subsequent trials weren't blinded.
Right, and that's been the confusion, that they call it, in some cases they call it double blind, and then here, at least in the US, we've seen people come on TV and give interviews that they got this vaccine and they feel there are no side effects or blah, 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 and then they're supposed to be enrolled in these clinical trials that are supposed to be blind. So then the question was, how did you know what you got? And then they said, well, because they didn't have an effect, therefore we early released them. So then you're really compromising your own data. And to me, again, that's not what I know of clinical protocols. They are pretty strictly written, and you're supposed to follow them. And yet there is just all this loosey-goosey thing going on that the very first time that when they announced from their phase 1 data, which was done in Germany with 60 patients, they jumped the gun and announced that it was 95% efficacious. Again, 3 weeks or 4 weeks before the trial was supposed to end. So it's just been very, very confusing, and it doesn't seem to be blind or what they say at certain points.
That would be really concerning if these are not blinded data sets.
Yes. Mm-hmm.
I didn't know whether with the RNA vaccines there's any evidence about transmission, whether it does reduce any transmission with this.
So I think that's a controversy that's also going on with so why do we not in some ways we should be sequencing viruses from people who get these breakthrough infections, but instead of as well as people sampling from general population who hasn't been vaccinated, what's happening to the virus and who is this virus coming from. But I don't think there is any data that's out there to say that. The other data that I have not been able to find is that when these mRNA vaccines are given, how much of the spike protein is made, where is it made, where does it get localized? There's none of that available. So I really don't know, what to make of it, because the other piece of it is that it is an RNA, it's a non-self genome, and it's going to evoke an immune reaction. So it could be just that you could be making it against all the immunity that you see is coming from just it being a non-self genome introduced.
Right. And I was also thinking that I guess there is some kind of a confusion or misperception that this vaccine, people are comparing it to the chickenpox vaccine. And whenever I have these discussions, they tell me, well, you had the chickenpox and the measles vaccine. But they forget that this vaccine has to be, it's not for life, right? You cannot just take it once and then you're done.
Right, and I briefly mentioned that there are two different kinds of viruses, DNA viruses and RNA viruses. This is SARS-CoV-2, flu, rabies, they're all RNA viruses.
Yeah.
Whereas chickenpox, measles, and those are DNA viruses. Those viruses don't mutate as often. Mm-hmm. So even if you look naturally when you get infected, and again, Mukta can attest to that, is that, and especially when you are much younger, you have a very important component of the immune system called thymic education. And that happens, which allows you to have long-term memory cells, not just T cells, but B cells. And so, and because these viruses don't mutate that often, naturally we make long-term antibodies and memory cells for these. But the viruses that mutate very often, like the RNA viruses, which is flu viruses and coronaviruses, the body is smart and say, if it's going to mutate, it's going to have to come up, our immune system has to come up with new ways to combat that. So there's no point in making really long-term immunity against it, because again, if it mutates, then it has to, if you've changed your face and you have to be recognized every few months, then you have to have a technology that will recognize that. But if you have the same face and you never age in that respect, then you don't need a newer technology.
Right. And I think the question that has been going around is that, should I take the vaccine? Should I not? Because right now what it looks like is that where there were thousands of people dying, and after the vaccine rollout happened, then the deaths have come down. So why not take the vaccine? So, I'm totally convinced I don't need to take the vaccine because I never take the flu vaccine. And like my husband also mentioned, we've been meeting his parents and other 80, 90-year-olds and never had any problem. So I think the confusion that in lay language people want to hear is that if I decide not to take the vaccine, how can I get that confidence, that the fear? I think a lot of problems have come about because of fear as well. So how can you as a scientist tell people that they don't need to be fearful? I know you did show all that data, like look at the number of people dying of heart disease. Even today, there are way more than people dying with COVID, and it's a media issue and it's the fear issue. And in India, and you can correct me, I think a lot of problems happened because people were not doing anything for, once you start the mucus buildup, you could do a lot of things to kind of stop it over there. But then there's a lot of over medication problems as well. So can you speak to that a little bit?
Multiple things. So there is one more question that I want to address, which is that I am not advocating it's your personal choice whether or not you want to take vaccine. But one thing to keep in mind is that if you have, for example, just diabetes alone, you have a certain chance of, let's say, having worse outcomes or dying from having that disease. But on top of that, if you have diabetes and you have heart disease, your chances of worse outcomes and mortality, which is or morbidity, it actually doubles. So when you have any two things wrong with you, your chances of having an adverse outcome or a worse outcome compared to a normal person who does not have any comorbidity increases. Unfortunately, globally, people have gotten unhealthier and unhealthier. In India, there are a lot of people with diabetes, a lot of people with heart disease. We saw those numbers. And so on top of that, if you get COVID, then, or if you get any flu or you get any of those other viral diseases, your chances of dying may increase. And then, like Sarah said, certainly in India, the medical system works very differently from the medical system the way it works here. So we cannot call in for antibiotics or steroids or other kind of medication. It has to be prescribed. If you had something, you would go to the hospital, and if you were not that severely affected, you would probably get triaged. You're not going to get treated, you're going to be sent home. But if you now can call in and take antibiotics by yourself, or you start self-medicating without really knowing what the root cause is, then you may inadvertently make yourself more vulnerable to secondary infections, which is what's happened in India. That a lot of people have gotten fungal infections, white fungus, black fungus, and they're dying from that rather than dying from COVID. So I do think that there is a little bit of that there in
right. And so I guess people, again, that question, how do we give confidence to someone who does not want to take the vaccine and she
so
or he
I'm
is being
going to
pressurized?
just look at more questions here, Sarah, and not talk so much about vaccine for now, because one of the other things that, like Pauline is pointing out, and actually you pointed out, is that because of our obsession with COVID or fear of COVID, a lot of people are not going to the hospitals or not seeking medical care for other diseases. So people are dying of other diseases, or they're not being given enough attention if they don't have COVID. So that's again a problem, and therefore it makes it incumbent upon us to be a little bit more responsible and say that we need to now go back and start looking at this profession in a more objective manner.
Yeah. Okay. Do you want to answer the question from Rakhi about the video that you've taken the vaccine and died?
Yes, I do not think that people are going to die from the vaccine. No, there is no truth in that. There's no such thing as it's basically. I haven't seen the video, but I've heard about it from many people, and I think it's completely bogus. No, you're not going to die from vaccines, or there is no conspiracy to kill half the population who's gotten vaccinated. I don't think so. But the question here is really what is the balance, and really I think in some ways some people don't have a choice. Some people have taken divergent pathways, whereas Pauline, because of her situation, has chosen to take the vaccine so that she can feel better at that she is contributing to the society by making sure that even if she got infected, she's not passing it along. But that's based on the assumption that the vaccine has actually given her enough antibody booster, and we don't know that. So if that's the case, then great. But on the other hand, people who've already gotten the virus and have gotten symptoms, they are just as or better protected than people who have been vaccinated. There was another study from London where they actually recruited. They had this a longitudinal study where they had patients from previous years, and they called them again, and they had people in that subset of patients who had flu virus, but not, and people who had taken or not had flu virus before. And then they analyzed their blood samples and found that the subset of patients who had flu virus, even though they had COVID, they could detect COVID, they did not develop symptoms for COVID. That means that even if you had flu, it was actually cross-reacting and protecting you from having COVID. So just keeping in mind that there are there's a lot of cross-reactivity, which we call polyclonal antibodies. So if you have one kind of infection and if the viruses are closely related, then your body will develop antibodies against not just one part of the virus. So it's like I showed you in that cartoon, the vaccines are only giving you spike protein. But when you get an actual infection, we don't know what part of the virus is or what different sections of the virus our bodies make antibodies to. So that means if the hand of the virus could look very similar to some other virus, then that means if you were ever infected with that virus, then your body could make antibodies against that virus and you may be protected. So with that rationale, I feel that if people who are healthy, who can take that hit, they should go ahead and take that hit. And that's also our control population, which will actually provide better immunity to the others.
And why is that message not going to the masses?
There's another question here asking, what about the Pfizer vaccine attacking the placenta? I'm not quite sure about what you mean.
I guess there are some reports. I guess there are some reports that they're referring to. I haven't read that, but
I think there's been some stuff on social media, like mainly WhatsApp and other social media, that people have been saying this. But I think both the CDC and I think the NHS have things on their websites that say that that's actually, there's no evidence for this at present, with the limited data that we have, that there's anything that affects fertility and or pregnancy.
Right, and actually quite an amazing organ. So I would be a little cautious about believing everything that you read on social media.
Right. So is there any precautions that you could suggest to people who are thinking of not taking the vaccine, or just want to kind of live their life normally and go about their day?
Sorry, I was reading the question. Okay. I missed your...
Well, no, I said, are there any precautions or preventions that you could advise or suggest to people who have decided not to take the vaccine and they just want to feel confident that, I'm not taking the vaccine and I'm still taking care of my health and I'm not infecting myself or others and I'm not being irresponsible?
Well, like I said, I think these are still, the jury is still out. There's a question from Irina asking that something similar to what you're asking, that if vaccine reduce hospitalization and death, an unknown impact on reduced transmission, why are we vaccinating all groups of population when older adults are more severely impacted? So basically saying something similar, yeah, I think we should, like I said, under any other circumstances, this is still under emergency use, right? So should we go ahead and immunize everybody not knowing what the outcome is, and given that what we know or understand about the immune system, that when you get natural infection, you don't need a booster. Have you ever heard of a booster from a natural infection? And the answer is no. So why are we not letting people who are otherwise healthy just go through with their life? And if you are so scared of whether or not people who've not been vaccinated, can they spread? I haven't been vaccinated, but I can tell you for over a year, and I have traveled and I have come in contact with other people, but I take precautions. When I meet strangers, I'm wearing my mask, and we do have the luxury that, at least in the Indian culture, you don't have to shake hands, so I can still go and say, I don't want to just do namaste or just fold my hands. So I think those kind of can take. So I don't know, Irina, does that answer your question? I don't know why are we forcing everybody to get vaccinated. I do think that we need individuals, and we need to really know more whether everybody who's gotten vaccinated, what's the percent that actually has antibodies. We don't know that.
Yeah, those messages are not coming out, and a lot of fear and panic. And I just want to kind of remind everyone that, this is a vaccine you will have to take every year, right? It's not, if you don't take it this year, you take it next year if you feel more confident about it.
So I have a question from Monica asking, can I talk about cytokine storm? So cytokine storm is, again, a word that's just thrown around. Any viral, bacterial, any kind of infection you get a cytokine storm. And you normally have the usual suspects. So you'll hear about TNF-alpha, you'll hear, depending on what happens, if you even cut yourself, you'll have some sort of an immune reaction. And that's why, so again, when you get infected with flu virus, you can have a cytokine storm. And that's not something that happens in everybody. You do get, meaning what they call a cytokine storm is when you have overactivation and overproduction of your certain kind of cytokines, which those cytokines are released because that's a signal to say, get rid of that bad thing, that infection. So once you get rid of it, it's supposed to come down to normal levels. And when it cannot come down, that's what you call like a cytokine storm. It remains elevated. And so your body is not being able to come back to what we call homeostasis. And that's what creates a problem. And that's what we refer to as cytokine storm in a way.
So, are there any other things that you would like to kind of mention in this that you have had people?
I just want to make sure that I'm going through all the questions.
Yeah,
and,
no, I looked at it. I think you've been pretty good with that. You've answered. There's a big, long one from Haris, and we answered that. So, yeah, the video about that has been answered. And whether people, I mean, there have been people who have. My question is, okay, so people have died after taking the vaccine, but then when asked, okay, so this person had taken both the doses and they died, and within the scientific community, what I've heard is that they start talking about the comorbidities that person had and said that, oh, that person had a heart disease and diabetes and that's why they died. And I feel that's just kind of going around in circles, that, okay, so this person had the comorbidities, but they also took the vaccine so that they would be better protected. But now, when they die, then we are attributing their death to the comorbidities that they were already living with. So I don't know, why are we going around in circles like that? And especially the scientific community, I'm a little disheartened.
Right, and I don't think not everybody who's died from vaccine or who's had thrombopenia or whatever other effects have had comorbidity, healthy people. But I think it's, again, you know, as far as I'm concerned, I would rather take the chance of naturally getting it and then seeing what happens. But that's unfortunate that happens. But that means that we need to study it more, right? So we need to have more safety studies. We need to know why in some subset of people we are having that reaction. And I think CDC is now going to focus on trying to understand why in a subset of people who get the vaccine without any other known comorbidities, why are they dying? I think they're going to focus on that. So it's the right thing to do. But again, that's if you were in that 1%, you know, do I want to take that chance? I would. For me, I would just rather take it naturally.
Yeah, like malaria. So, you know, people get malaria and some die, some have side effects, and some come out of it. So I think there's also a misconception among people that had I taken the vaccine, I would have had less side effects.
So we talked about, I had a slide about COVID long haulers, and if I've known people in India, including my mother, who had chikungunya, and I think she suffered from that, the side effects and fatigue from it for 2 to 3 years after she had people who get mono. They suffer from fatigue and other issues for years. So to say that it's only happening with COVID-19, or you could take the vaccine and it may not happen, not true. And more research is coming out that people, there is also the sex difference, right? So more men were dying from it, and women seem to be protected. However, there is a subset of women who have certain comorbidities, such as polycystic ovaries and things like that. Their COVID-19 symptoms are worse than a normal man's symptoms, and they suffer from it for much longer than any other normal person would. So we are still learning, and new research and new data are coming out as to what are the other things that what permutation and combinations make this better or worse. And that's why I feel that if we let this virus go through its natural cycle, the cat's out of the bag, if more people who are healthy get it, then they'll hopefully be less pathogenic, less virulent, and less able to cause damage than
and masking it. I think that's the right term. You said, let the cat come out of the bag. And this has been studied way more than we should have studied malaria, I think. Right? We haven't reduced the incidence of malaria in all these years.
And the easiest way to reduce malaria is actually use the mosquito nets.
Exactly, and same here.
So sometimes it's not always technology. I mean, despite all the billions for, and that's a different topic about genetically modifying mosquitoes. My biggest worry with that kind of science is that let's be very clear. These parasites and things have been around for millions of years before human race came around. So if you now change the host, meaning you mutate the mosquito, the parasite, which is Plasmodium, which causes malaria, can find another host.
Exactly.
It's not going to go away. Now, just imagine right now the host is confined. The mosquito that causes malaria is only confined to Africa and Asia. And if Plasmodium jumps host or goes to or finds another host, and it now affects worldwide, look at the devastation you're going to cause. So having said that, there is in evolution you find that people who live in the sub-Sahara region where malaria is very predominant, and in India, they also have something called sickle cell anemia, because Plasmodium cannot grow in sickle cell red blood cells, and therefore those people are naturally resistant to having malaria. So in those times, either you die from malaria or you get sickle cell and you never get malaria, but you will die from another disease. So it's again, give and take. It's a good example. That's what we have to just learn, that we cannot, there is a give and take. So same with COVID-19. We just have to come to terms with the fact, and actually it's time to move on.
Right. And so I guess if no one else has a question, my last would be, so how do we improve our immunity?
Well, I think you have several blogs about that.
But you know what I've been hearing is that...
I will direct people to that.
Oh my goodness. No, I've stopped writing, because when I tell people food is medicine, I'm told that it takes a long time and food is really not helpful. So I've stopped writing about it.
Well, I think doing a lot of gut-brain research that I do, you are what you eat. So I think it's very true. You can choose wisely to eat. We know even with type 2 diabetes, you can really control it with food and exercise. So that's, in fact, in UK has done the biggest studies following, and they've given them diet control, and now they've gotten more than half of their enrolled patients actually off of insulin just by doing diet control. They're not even doing exercise. Right. So I do think that diet has a huge role. And as somebody who studies stress, trying to have a positive attitude, which you will reduce your stress and boost your immune system, because stress is downstream regulator of that is cortisol, which has, and again, a very paradoxical effect on the immune system and on your health. So I think there are a number of things you can do, and which is to boost your long-term immunity, which includes good habits, good diet, good food,
sleeping,
and everything in moderation.
Yeah, I think people are forgetting the importance of just sleeping on time between 10 and 11 p.m. and waking up early, after sleeping for 7-8 hours. And in fact, Canada has just issued something called the Canadian Sleep Guideline. And it's basically if you sleep longer than 8 hours, you're actually affecting your heart, and you actually become more prone to heart disease. So that's also We can't just keep on randomly sleeping any odd time and expect to be healthy and immune. And I keep saying that, reduce the mucus. Like, you can take some precaution every day. Just do saltwater gargles. It's not going to hurt you. It's going to clear your mouth, have a better breath, and you might improve your immunity. Is that a good thing to do?
Most of these things, again, work in subset of patients or people. Yeah. So, probiotics or, it's very hard to, things that are, I think, very difficult in some ways, because your microbiome, you inherit from your mother, most of it. And again, when you come through the birth canal, that's when you pick up your mom's microbiota. Right. And so if it's a healthy microbiota, and some of it comes from breastfeeding, they are definitely, and so probiotics in India, people used to eat yogurt or dahi with their meal all the time, homemade dahi. Again, there are different bacteria in yogurt that are sold here. In India, the predominant species was lactobacillus, which is very actually helpful, but that may not happen with other kind of yogurt or synthetically or artificially made things. So there is a lot that we can do.
But the probiotics is something that we shouldn't aim to just at random pick up anything from the store and start having it. You really need to know what you're taking, why you're taking, for how long you're taking, and is your system really ready to start taking something like that. But natural probiotic is, yogurt. You can definitely choose that, and that may not harm you. And or even kimchi, like fermented vegetables. Those are things that are always good. So whole foods, basically I circle back everything to whole foods, plant-based diet, sleep well, exercise regularly, sweat it out, get sunlight in your hair, on your skin. I don't think you can go wrong with that.
I think now this is a topic for another discussion.
So with that, thank you so much. Unless someone has one very last burning question.